the way through talking about DNA structure, so I'm going to finish up the last couple of slides on this particular topic, and then I think I will have us remake our list of things that we understand about DNA structure so that we can make sure that we've hit sort of all the major pieces in there. Right, so I think I ended, did I end on this slide? Is that right? The next one? I think one thing that I actually didn't talk about was spacing and sizing of the nucleotides, right? So I don't think I mentioned these numbers. So in our 10 base pair 360-degree turn, that is going to have a standard length along a strand of DNA. That standard length is going to be 3.4 nanometers. So for every 10 base pairs in length that we have in a sequence, or in any piece of DNA, we can determine how long this piece is when it's not compacted. So it's not kind of crunched down into that chromosomal structure that we're so familiar with. That means that each of the nucleotides takes up 0.34 nanometers. If I said we have a strand of DNA that is 100 nucleotides in length, how long is that strand of DNA? You should be able to tell me. So 100 nucleotides would take up what would be what length?
40 nanometers?
34 nanometers.
Oh.
Right, 34, so you just multiply that by 10, gives you 100. So 34 nanometers. You can also, if I said we have 126 nucleotides, how many 360-degree turns would we expect? 126 nucleotides in length. How many? 12.6 complete 360-degree turns. Does that make sense to everyone? 10's a nice number. Thank you to our DNA for making this a base 10 system. It works really nice. Okay. Oh yeah, I did use this, I remember now. So in addition to our hydrogen bonding between our bases, which gives our helix stability, we also have a specific orientation of base stacking, and what I mean by that is the bases are actually flat to each other. So if you envision a nucleotide ring structure, you can think of that as being like a plate, right? And so normally when we draw it out, the plate is like standing up, it's like facing you. When they actually line up and form DNA, they are stacked like you put plates in a cabinet. So when you're putting away your dishes, and you put a plate on top of a plate on top of a plate, right? Everybody know what I'm talking about? That's how it's true though, that's how we stack our DNA molecules, or our nucleotides. So they're not like this. That's not very stable, right? So when you're putting away dishes, you don't put the edge of a plate on top of the edge of a plate. That's not going to stay like that, unless you're a magician. Or you have a really cool system of plate stacking. It also takes up a lot of space. Right? If we flatten them out, we can stack quite a few of them in a very stable structure in a very limited amount of space. Right? So when we're making DNA, we kind of have to think about how is this molecule going to stay together and function properly, and how are we going to fit all of this information into the nucleus of the cell. Right? And so that's sort of a two-piece system right there, where if we stack the bases properly, we're taking up less space and we get increased stability. And then I'm actually going to go to the next slide to show our grooves. I think it's a little bit clearer. So this is sort of almost a, I would say two-dimensional, but it's a three-dimensional representation, or it's intended to look like a three-dimensional representation of a DNA double helix. When we have our helix twisted and represented like this, we can see that, so what part of the DNA is this? And this. And this. Yeah, so this is our sugar phosphate backbone. Okay, most of the exterior surface of our DNA molecule is going to be sugar phosphate backbone. Remember I said that our nucleotide bases are polar, and so they are hydrophobic. So what is the inside of our cells mostly comprised of? The body in general is in large part water, right? So inside of our cells are just sacks of water. And then we have some other things kind of shoved in there. DNA is one of those things. It's an aqueous environment, so it's surrounded by water. It wouldn't make a lot of sense if our nucleotide bases were exposed to the water because they would repel each other. Right? And that doesn't make for very good living conditions. So when we have this sugar phosphate backbone exposed, we are now sort of protecting that hydrophobic inside from the aqueous environment. Prevents things from getting in, and it also keeps sort of a stability, pulls those molecules together even more. And that's part of why the structure looks the way it does. There are grooves or like slight openings into the nucleotide bases, right? So there's a little bit of a gap between this rung of the backbone and this rung as it twists around. Small gap here, that's called our minor groove. On the other side, there is a major groove. Okay, so this would be like in a 360-degree span, you're going to have a minor groove where the backbones are very close together, and then you're going to have a major groove where there's a little bit of distance between the two backbones. So we do have some accessibility into the nucleotide sequence. When are we going to need access to those nucleotides? What processes within the cell require access to DNA?
You gotta copy it in the interface.
Okay, so replication, right? When we replicate DNA, which we'll talk about next week, we have to have access to our template strands. We can't make a copy of this molecule without having access to the nucleotide bases to figure out what order they're in. Okay? When else do we need to have access to DNA? Transcription. Translation would be mRNA. And so transcription is when we're making, so it's going to be the first process in our central dogma. Oh, maybe the first process in our central dogma, which is how we go from DNA to protein. And actually, now that I've mentioned it, let's talk about our central dogma. Now, I know for a fact that Dr. Irvin uses this terminology. He is introducing molecular concepts in introductory biology. Sorry. What is our central dogma?
DNA to RNA.
Okay.
RNA to protein, protein to phenotype.
Okay. You said DNA to RNA?
Yeah.
Okay. Yeah. I'm going to make an mRNA. Is everybody familiar with this? Well, if you're not, I suggest you write it down. Put a star by it, maybe. Remember it for your next exam. Yes, this is important, right? This is actually what we're starting to talk about here. So we're talking about DNA right now. In order for DNA to persist in our cells, we have to go through this process called replication. We have to make copies of DNA to keep copies of DNA. This arrow represents what process? So DNA to mRNA is our transcription. To get from mRNA to protein, translation. And then protein to phenotype would be basically expression. Which we're gonna stop our journey at protein. Although we have already talked about how genes influence phenotypes. So we're filling in the middle pieces now to how that happens. We kind of just went from here to here to start. Okay? So can everybody see the relationships between all of these molecules? Right, so in order to get DNA, we have to replicate DNA. In order to get mRNA, we have to use DNA as a template through the process of transcription. Without DNA, we never get proteins. Without DNA, our phenotypes are non-existent. With a change in our DNA, our phenotypes change through all these processes. So, the reason that I'm bringing these up now is because these two processes, we have to have access to our DNA sequence. So, the way that we gain access to those nucleotides is through these major and minor grooves. So, most of the time, these major and minor grooves are too small for most molecules to sort of access or to get in and cause damage or changes to the nucleotide sequence. But the enzymes and the cellular machinery that allows us to do these two things are designed to fit into the major and minor grooves at the initiation of the process, and then open up the DNA. Okay, so these grooves are necessary in order for these things to take place, or in order for our central dogma to work. Yeah, so here you can kind of see the major and minor grooves. So this is the B form, so there are isomers basically of DNA, so it's the same molecular structure, but they are topographically different, so they have different structures. This is our common form in life, so almost every cell and every life form uses B DNA as the primary type. So if we look here, blue and yellow, those are going to be our backbones, those are our sugar and phosphate groups. Gray are the nucleotides. So this is in that double helix conformation. This, what type of groove is this? Minor, major, right? So you can see the difference between the size of those. And obviously the major grooves are larger, so you're going to be able to get enzymes and proteins and things wedged in there a little bit better. Okay, so there are other forms of DNA. I'm not really going to talk about A-form DNA. I don't think that it ever really exists, as far as I know. If it does, it's very, very rare. Z-form DNA is very similar to B-form DNA, except for that this is a left-handed helix. So remember that structurally our DNA rotates to the right, clockwise, as it moves out from your body. And I'm just imagining strands of DNA like shooting out of you, spinning to the right. So if it was left, it actually changes how many nucleotides are within a single turn. So here we have, so these are actually molecules of the same nucleotide length, so they have the same number of nucleotides. If you notice though, the Z-form DNA is longer. That's because we have 12 base pairs per turn instead of 10. Okay, so it's spread out more. It changes the overall relationship between the nucleotides. So zDNA does occur. If we actually just look at the backbones are different. So these backbones on the zDNA actually forms like a zigzag instead of perpendicular lines, right? These two backbones essentially never intersect and they're always parallel to each other. Here they actually form like these interesting zigzag, it's a little bit hard to tell here, but like this zigzag patterning. And the bases also are not oriented at the same angle to each other in zDNA. So we do find zDNA in some cells. Not always entirely sure why, but there do seem to be some conditions that favor the presence of zDNA, and so those are sort of listed there. And it can at times be involved in transcription and chromosomal structuring, although again, most of the time we see zDNA. So zDNA is still a rare sort of variant. And bDNA can actually adapt to a zDNA form under certain conditions. So we'd start with bDNA, and then it would change conformation if we had low internal salt concentrations and lots of cytosine methylation. During what process do we get methylcytosine or methylated cytosine? We talked about it in chapter 5. That's like my favorite process ever. I don't think you guys talked about it very well. Epigenetic markings. Yeah, so like how your environment influences expression of your genes. We talked about it a little bit. I know. Did we talk about how like traumatic events that occur in a grandparent's life can actually cause epigenetic changes in their like grandchildren? It's crazy. Craziness. All right. So just quickly on RNA structure. So as you can see up there, as we get into transcription and translation, we are going to be talking about RNA. RNA in general is much less stable than DNA. So has anybody done RNA work? Worked with RNA? Okay. That'd actually be a really good cell molecular experiment to do. So you'll see when we are working with DNA in lab, DNA is really robust and it's really hardy. So we can extract DNA from things that are long dead, things that are very old, things that have been exposed to very high temperature. Those conditions break down most other things, but DNA is so stable that it persists, right? And so we can use it, right? So if we think about these, has anybody, have you guys had Dr. Hammond? Most of you? Have you, okay. Did you talk about like phylogenies with like Neanderthals and stuff like that? Okay, so in that case, Neanderthals as a species stopped existing thousands of years ago, but we can still extract viable DNA from Neanderthal bone. That's crazy. Yeah, it's awesome. RNA degrades within minutes or hours of a cell stopping function. So once cell death or organismal death occurs, RNA, it's so fragile. If you heat it or cool it, like you take it in and out of a freezer one too many times, it's gone. If you are looking to determine gene expression, what you would do is you'd actually sequence mRNA, the number of mRNA copies that you have. That tells you how many proteins are going to be produced. You have to extract tissues or cells from a living organism and immediately put it in, it's called RNA later, but it's a reagent that stops RNA degradation. Otherwise, your sample's gone. I did RNA work once, and I did not read, not done work once, but I was in an RNA style lab for a while, and I thought it was doing super awesome, but I forgot to add one of the components to my RNA extraction. And I was like, oh, it'll be fine. It was not fine. And I was doing mosquito work, so we had to hatch them from eggs, rear them to adulthood, wait until the females are mature, and then dissect out ovaries. So it's like a 6-week process to get the samples to extract the RNA. And I messed it up in 1 second of just not adding the right thing. I'll never forget that again. So yes, RNA is very unstable, especially compared to DNA. It looks a lot like DNA in many ways, right? So we have very similar nucleotide composition where we have A's and C's and G's, but remember that instead of thymine, we now use uracil. Yes, is uracil a purine or pyrimidine? Pyrimidine, yep, good job. We also have a different sugar. So our ribose, the way that it's different from deoxyribose pentose sugars is this presence of this hydroxyl group on the 2' carbon. Listen to me use, that is right. Couldn't tell you. Hydroxyl groups, yes. On the 2' carbon that is not present in our deoxyribose groups. My chemistry professors would be so proud of me. I'm sure they did not think I would make it here. Okay, they aren't nearly as long. Why are they not as long as like a chromosome? Chromosomes are millions of base pairs. Why are RNA molecules usually only hundreds or thousands of nucleotides in length? So let's just say we're talking about mRNA. When we are producing mRNA to make proteins, do we copy the entire chromosome? No, we copy a gene region, and those gene regions are usually a couple hundred to maybe a couple thousand nucleotides in length. So RNA molecules are shorter. They are almost always one strand, okay, like this. We do sometimes see really interesting secondary structures, usually still in just one strand of RNA, right? So we get like these loop structures like this, and then even, does anybody know what this is? A what? A ribbon? I'm sad now. Or as my daughter would say, that makes me so sad.
Oh,
Yeah, but what is that? What is that ribbon of?
It's not DNA.
It's RNA. Nobody knows what this is?
No, it's just a.
This is a tRNA molecule. tRNA is how we make proteins. Oh, it's so good that we're going to be talking about translation in 2 weeks. Okay. Yeah. But this is all one piece of RNA. It's just wrapped around, right? You can follow it all the way back to the other end. So it's one strand, but it has this really sort of highly complex folding pattern, and that's what allows tRNAs to carry amino acids and make proteins. Okay. I feel like there was something else I wanted to say about RNA. I guess not. Alright. Cool. Any questions? Okay, so I'm going to move into the next part, and then when we come back on Monday, after you've had 2 days of not thinking about DNA structure, and then I'm going to make us make a list of all of the DNA structural components. All right. You guys have fun plans this weekend? I heard there's trick or treating.
We have our first scrimmage.
Ooh, nice. Here?
Nope, Stevens Point.
Oh,
Our first game is the 6th.
okay. Nice.
Here, you like?
Anyone else fun plans? Oh, weddings are fun sometimes.
They've been skating for like 6 years. We were all surprised that actually got made.
It's a long time.
That's your party.
Oh, that'll be all right. So, now that we know all about DNA structure, I started the thing. All right, so now that we know all about the structure of sort of individual strands of DNA, we're going to talk about how we take those strands of DNA and compact them into our chromosomes. Right? So, as a reminder of why DNA sequence is important, it allows us to synthesize or make RNA molecules. Without that ability, we could not make proteins. Usually, if we can't make a protein, that changes our phenotype. And sometimes, as we discussed with Mendel, that phenotypic change is not a big deal. Sometimes that phenotypic change can be lethal. Again, without DNA, as we just talked about, we cannot replicate chromosomes. If we can't replicate chromosomes, we can't make new cells through mitosis, and we can't make gametes through meiosis, right? Both of those processes require us to replicate chromosomes first. Particular or specific DNA sequences allows us to segregate our chromosomes, and I think we touched on this a little bit when we were doing mitosis and meiosis, but maybe I wasn't super clear on it. There are specific sequences of DNA at those centromere regions that are what allow the microtubules to connect and then pull apart those chromosomes. So without a centromere, a microtubule is never going to attach to a chromosome and pull it in one direction or another. Okay, so without a centromere, we simply cannot segregate chromosomes, and that centromere is a highly specific sequence of DNA. And then the last piece, which is what we're going to primarily focus on in this chapter, is we use DNA sequence to compact chromosomes. So far, and I'll show you this image again later, but so far the way we've talked about DNA has been like this. Just our simple double helix thread or strand. I guess we've also talked about DNA in this structure, in our chromosomes, right? In our sort of in meiosis and mitosis style chromosomes. There's a lot that goes on in between that allows us to get from here to here. It's all about how we pack down those chromosomes and make them fit into the nucleus of the cell. Boom, didn't even need to talk about it. Central dogma. It's a nice thing. Okay, as we talk about all of the pieces of our central dogma, so we talk about replication and transcription and translation over the next 3 weeks, we are primarily going to focus on bacteria, which are they eukaryotic or prokaryotic? Prokaryotic. Okay. What's the difference between a prokaryotic cell and a eukaryotic cell? Okay, prokaryotic cells don't have a nucleus. What else? They lack membrane-bound organelles. What else?
Unicellular and multicellular.
Say that again?
Unicellular and multicellular.
Typically, they're unicellular. What about their chromosomes?
There's no variation.
Okay, so it's a spaghetti chromosome.
They're not in the nucleus.
They're not nucleus bound. How many do they typically have? One. Okay, and what is its structure? A circle. Okay, this is very different from eukaryotic cells. Eukaryotic cells are much more complex. There are still a lot of things or a lot of parts of these processes that we do not know and do not understand. So I simply cannot teach you transcription in a eukaryotic cell when we only know vaguely some of it. Okay? So we do know, it's not like we don't know anything, but we use prokaryotic cells, and in particular bacteria, because they're a really nice model organism, but we use these as a way to talk about the processes that are ongoing, and then I will discuss what we do know about eukaryotic cells sort of in this framework. Okay, so it's important now to sort of understand that as I'm talking about what's happening in prokaryotes, and that is similar in eukaryotes, but it's not identical. And so we have to know a little bit about our prokaryotic friends. So, maybe I should write purple. Last time I wrote purple, okay, so let's sort of list or look at this chromosome that's in our bacterial cell and make sort of a list of features. So we already said that usually they have circular chromosomes. That's not working well. Circular chromosomes. The end of this came out. Okay, usually they just have one chromosome. What else do we know about their chromosomes? How long are they? Longer or shorter than a eukaryotic cell's genome? Yeah, so usually this chromosome is a couple million of base pairs in length or nucleotide. So I'm gonna, anytime I'm abbreviating nucleotide or base pair, which are the same thing, a base pair is a nucleotide or a nucleotide is a nucleotide, I'm gonna abbreviate it as BP or NT, just so you know. I'm not gonna write out base pair and nucleotide 3 times. You also may see that in the lecture at once. All right, in that million or millions of base pair, how much of that is gene coding sequence? How much of the human genome is gene coding? So how much of the human genome actually results in the production of proteins? A lot? A little? A medium amount? A little. Yeah, we have tons of non-gene coding sequences in our genome. We have 300 million nucleotides. And we have like maybe thousands of genes that are each 100 to a couple thousand base pair elements. We have tons of DNA sequence that does nothing for it. Well, that's unfair. That is not directly coding for proteins. Okay? In a prokaryotic organism that just has one chromosome, we actually have hundreds of gene coding regions. Of this million or couple million base pair, it's mostly protein coding or gene coding. Okay, so in a bacterial cell, they have fewer chromosomes, their chromosomes are shorter, they still have a lot of genes within that chromosome, which simply means that they don't have a lot of non-gene coding sequence. Right, so if we sort of look at this example, everywhere that we have black lines going around are going to be genes. Anywhere that there's red lines or sort of this double helix structure, those are what we call intergenic regions. So those are just segments of DNA between genes that don't actually code for anything, or repetitive sequences, which again are non-coding regions. But you can see that they're interspersed, but they're fairly short. And there's lots, comparatively, there's lots of gene coding sequence. If we looked at a human chromosome, it'd be like gene coding and then like huge big stretches of non-gene coding, and then gene coding, and then, right, so we get lots more non-coding compared to our gene coding regions. All right, I think those are sort of the major things that we need to know. And this is a prokaryotic cell, right? So this is an example of a bacterial cell. And you should note that this chromosome is sort of spread out throughout the cytoplasm, cytosol. You guys learn the difference between those things? Cytosol and cytoplasm. Okay. I was making fun of my husband for being real picky about that, and I was like, who cares? It's the same thing. It's not the same thing, but we pretend it is. Okay. Anyways. All right, so even though we only have one chromosome in our bacterial friends, that chromosome still has to be packed down to fit inside of that cell. So here is a bacterial cell. It's a bacteria. All of this stringy stuff around it is its chromosome. So the chromosome has been taken out and sort of laid out around it to show how much DNA is actually compact and put into the center of this cell. So that's a lot of DNA, a lot of yarn to sort of ball back up and put into that cell. And this is another way to look at it. If this is the size of an E. coli cell, obviously this is larger than the size of an E. coli cell, but if that's what an E. coli cell looks like, this line around this slide would be the amount of DNA that has to be wrapped up and put inside of that cell. So proportionally, I think it's impressive. Maybe you're not impressed. So the way that bacterial chromosomes are compacted is through the formation of what we call microdomains. So microdomains are these big loop-like structures, which you can't really see here, but, and through supercoiling. So on top of already being a double helix structure, which in and of itself packs down the length of a DNA molecule, DNA is also supercoiled, so it is twisted in addition to its already twisted structure. There are actually sort of two reasons that we twist DNA. When we twist DNA, it allows it to pack down, and so sort of as an example, if you take a rubber band and you hold either end and you twist it up and then you sort of relax, that rubber band naturally folds in on itself. I know that you all are maybe too young for this one. Yes? Corded phone. Well, I guess there's still corded phones in offices and stuff, right?
I still have a house phone.
Yeah.
My mom refuses to get rid of it.
We just took, my parents do too, we just took the jack out of the kitchen in the house that we own. So nobody's ever gonna use this again. But yeah, so this would be an example of supercoiling, right, where that twists around itself sort of naturally. Okay, we can twist DNA in two ways. So we can twist it in the direction it's already twisting, right? So we can add another right-hand turn to it. Or we can untwist it, or twist it in the opposite way that it's already twisted and twist it to the left. So if you were to guess which direction our cells or a bacterial cell twists its DNA, what would you think? Okay, to the right. So you think that to pack it down, you're going to twist it further in the direction that it naturally twists. Amber's saying no. Yeah, so interestingly enough, the cells are actually going to under-rotate or underwind the DNA by twisting it to the left. Now that doesn't make inherent sense to a lot of people, but it actually accomplishes the two purposes that we're looking for. It compacts the DNA because we still get sort of this secondary folding of the molecule, and that loosening of the turns actually allows us better access to particular regions of DNA that are necessary to start replication and to start transcription. Okay, so negative supercoiling is going to be the predominant type of supercoiling that we see in a DNA molecule. So we actually twist it to the left. When we do that, we're still creating tension. We're still packing down the molecule. And we're still causing this, these certain locations to sort of start opening or pulling apart a little bit so that we can wedge our replication and transcription molecules in there. So here's an example of what might happen during negative supercoiling of a bacterial chromosome. So when we create that negative tension, it can cause the double helix to start pulling apart. And again, that's going to allow us easier access to some of those nucleotides where we want to actually start replication and transcription and translation. sometimes we have supercoiling where we don't want it, or we get positive supercoiling where we want negative supercoiling, and it's changing sort of what the DNA is supposed to be doing in a cell. The problem is when we have supercoiling occur, it can be difficult to undo, because to undo supercoiling, we have to actually break or cut both strands of DNA, separate them, untwist one half, and then put them back together. Right? That's kind of a tricky process. We don't, in general, it's usually frowned upon to create double-stranded breaks in our DNA, because that opens up the possibility of error when we join them back together, or we don't join them back together. There is a particular enzyme that's known as DNA gyrase, or DNA topoisomerase 2. I would possibly encourage you to think of this term, although if gyrating of DNA makes sense to you in terms of untwisting things, because gyrate means to like twist. So untwisted. But anytime we have different structural types of DNA, so we have two strands of DNA, one of them is supercoiled and one of them's not. Those are called topoisomers, right? Where they are the same molecular composition, but different structural compositions. So they're topographically different, but they're still isomers because they're still the same thing. So the way DNA gyrase works is it can actually put negative supercoils into DNA, or if DNA is positively supercoiled, it can go in and undo that positive supercoiling, so we're back to the preferred either standard uncoiled DNA or negatively supercoiled DNA. And then I think I have, yeah, so this is what DNA gyrase or topoisomerase 2 does. So it has 4 subunits, 2 big subunits on the bottom, which are A subunits, and 2 on the top, which are B subunits. It gets a little bit complicated at this point, but we take our strand of DNA. It is held in the lower jaws or in the A subunits and then wrapped around the B subunits. We then cut it, so we make a double-stranded break. Meanwhile, both pieces are being held onto by this molecule to keep them from flying out. There's basically a big clamp that comes in and takes one half of the double strand and untwists it, and then we put them back together. I know that was very helpful. Oh, I have a video. Let's see what this looks like. Hopefully it's exciting. Double-stranded break. See how it untwists? of topoisomerase 2, okay, which is going to undo positive supercoiling for intracellular.
She'll be back.
All right.
Yeah.
Have a good weekend. If you're celebrating Halloween, have fun. Don't get too crazy. I want to see everybody on Monday, so.
I'm the blue one.
We're having a party at my bar then too. I'm not gonna be there. I'm here in the morning.
Our first two groups are only like 3 people each, and then the third group is like 12 people. Everybody signed up for these?
Raquel, why are you back there?
Yeah, I was like, where the
Oh.
fuck?
Gotcha.